This video is about Document 5 Oogenesis in Chapter 1 Basic Mechanisms of Sexual Reproduction, and at the end of it the student will be able to define oogenesis and indicate its site, name the different cells and tissues involved in oogenesis, explain the different phases of oogenesis, and finally explain the evolution of follicles during a menstrual cycle. First, oogenesis is the process that leads to the formation of haploid female gametes from diploid stem cells. It is a discontinuous process that does not end in the ovary. So, what are the different stages of oogenesis and how do follicles evolve during a menstrual cycle? And we will see later what are follicles. Now, let's study the histology of ovaries. If we examine a section of an ovary after puberty, we observe structures called follicles of different sizes, and these are the different types of follicles. While if we examine the ovary before puberty, we only find the smallest type of follicles or primordial follicles that are at the origin of the other types. Basically, a follicle consists of an oocyte surrounded by many layers of follicular cells. Starting from puberty, primordial follicles develop by getting more layers of cells, and we can see this in this document. They become primary, then secondary, then cavitary follicles that include a cavity. After that, the cavitary follicles turn into mature or Graafian follicles that undergo ovulation, and they are not presented in this document. Ovulation is the expulsion of the oocyte from the Graafian follicle into the fallopian tubes. The part of the follicle that remains in the ovary is called ruptured follicle, and it turns into the yellow body. Then the yellow body regresses in size to give the white body. Now, this document shows the structure of the mature follicle. Of course, each part has its specific role, but we are interested to the internal theca and the granulosa cells that secrete the female sex hormones, the estrogens. Now, let's start by studying folliculogenesis. First, what is folliculogenesis? Folliculogenesis is the development of follicles in the ovaries. This is folliculogenesis. Okay, 6 to 7 million primordial follicles are produced during the embryonic life after 20 weeks of pregnancy. Most of them degenerate during the remaining embryonic life and childhood, so that at puberty the ovaries only contain around 400,000 primordial follicles. Starting from puberty and monthly, around 500 primordial follicles start developing to give 3 to 4 months later 10 to 15 cavitary follicles. Of course, the primordial follicles become primary, then primary follicles turn into secondary follicles. After that, secondary follicles give the cavitary follicle. This process, which means the transformation of primordial follicles into cavitary follicles, need from 3 to 4 months. When cavitary follicles are obtained, the girl becomes ready to have a menstrual cycle. This is the menstrual cycle, and as you see, at the beginning of it, there are follicles with 5 millimeter of diameter that corresponds to cavitary follicles. And as you see, the menstrual cycle is divided into two phases separated by ovulation. The first phase is called follicular phase that lasts for 14 plus 3 days, which means that lasts for 11 to 17 days. The second phase is called the luteal phase that lasts for 14 plus minus 1 day, which means 13 to 15 days. The follicular phase starts with menstruation or bleeding for few days. Around the middle of the follicular phase, one of the cavitary follicles becomes mature or Graafian follicle. Few days later, ovulation occurs. After ovulation, the ruptured follicle turns into the yellow body. Then the yellow body turns into the white body at the end of the menstrual cycle. To be noticed that if fertilization takes place, the yellow body will be kept for around 3 months, and this stops the menstrual cycle. Now, let's study the oogenesis or formation of haploid female gametes. As you see in this document, oogenesis includes 4 phases. The first one is called multiplication. The second one is called growth. The third one is called maturation, or it is a meiosis. And the last phase is the differentiation. First, what happens during multiplication? The mother cell of oocyte is called oogonium. Oogonia is the plural name of oogonium. One cell is called oogonium. So, oogonia undergo successive mitosis to increase the number of oogonia. And we know that mitosis conserves the number of chromosomes. So all oogonia have 2n chromosomes. Multiplication occurs during the embryonic life and not after puberty. The second phase is called growth, and it consists of cellular development by storing certain substances, and the volume of the cell increases, and at the end of this phase the cell will be called an oocyte. The number of chromosomes didn't change in this cell, so it has 2n chromosomes. Now, we should also say that growth is done during the embryonic life. The first phase is the maturation or meiosis. The oocyte 1 obtained at the end of a growth starts meiosis, but it stops at prophase 1. This is the first arrest of oogenesis, and the completion of prophase 1 is done also at or during the embryonic life. Now, after puberty and few hours before ovulation, the oocyte 1 already blocked at prophase 1 completes the first meiotic division to give two unequal cells, the first polar body and an oocyte 2 with N chromosomes. And we know that meiosis 1 consists of separation of homologous chromosomes, so the oocyte 2 or secondary oocyte has N chromosomes with 2 chromatids. Now, what happens? This oocyte 2 starts the second meiotic division and stops at metaphase 2. This is the second arrest of oogenesis. Now, ovulation occurs and the oocyte 2 blocked at metaphase 2 passes from the Graafian follicle in the ovary to the fallopian tubes. Now, if fertilization doesn't take place, the oocyte 2 degenerates and oogenesis won't end. On the other hand, if fertilization takes place, the oocyte 2 blocked at metaphase 2 completes the second meiotic division to give the second polar body and an ootid with N chromosomes of 1 chromatid, because the second meiotic division or equational division consists of separation of homologous chromatid. So the ootid marks the end of the third phase or meiosis. Now, the last phase of oogenesis is called differentiation, by which the ootid differentiates, evolves to become an ovum. And when the sperm cell and the ovum unite, we obtain the zygote having 2N chromosomes. Concerning the two polar bodies, their role is to reduce the quantity of chromosomic material in the dividing cells so that we obtain haploid gametes. Also, their formation reduces the number of gametes obtained.